Herkese merhaba. Bugün 3 Haziran 2026 Çarşamba. Nano Open'da değerli bir konuğumuz var. Koç Üniversitesi KUTTAM'dan Doçent Doktor Safacan Kölemen. Hoş geldiniz.
Merhaba, hoş
Safacan,
bulduk.
Safacan Kölemen, sunum koordinasyonunda yürütülen ve kanser tedavisinde yenilikçi nano-biyoteknoloji temelli terapötik yaklaşımların geliştirilmesini amaçlayan nano-biyoteknoloji programı süpervizörleri arasında yer alıyor. Bugün yapacağı konuşmanın başlığı The Rise of Chemiluminescence Next Generation Probes for In Vivo Imaging. Webinar İngilizce olacak. Mikrofonu kendisine aktarmadan önce hakkında kısa bilgi vermek istiyorum. Doktor Kölemen lisans eğitimini 2008 yılında Bilkent Üniversitesi Kimya bölümünde tamamlamış ve aynı bölümde yüksek lisans derecesini almıştır. Doktorasını ise 2014 yılında Ulusal Nanoteknoloji Araştırma Merkezi UNAM bünyesinde Bilkent Üniversitesi Malzeme Bilimi ve Nanoteknoloji alanında tamamlamıştır. Ardından Kaliforniya Üniversitesi Berkeley'de doktora sonrası araştırmalar yürütmüş ve burada floresan moleküler sensörlerin geliştirilmesi üzerine çalışmıştır. 2017 yılında Koç Üniversitesi Kimya bölümüne katılan Doktor Kölemen, organik kimya ve kimyasal biyoloji laboratuvarını kurmuştur. Araştırmaları fototerapi için tümör seçici ilaç adayları ile biyogörüntüleme uygulamalarına yönelik floresan ve kemilümülisan ajanların tasarımı üzerine yoğunlaşmaktadır. Doktor Kölemen kariyerinin erken döneminde birçok prestijli araştırma ödülüne layık görülmüştür. Bunlar arasında BAGEP, TÜBA-GEBİP, ODTÜ Parlar Vakfı Ödülü ve TÜBİTAK Teşvik Ödülü bulunmaktadır. Ayrıca ACS Central Science ve ACS Biomed Chem dergilerinin editoryal danışma kurullarında görev yapmaktadır. Hocam tekrar hoş geldiniz. Ekran paylaşımını size devrediyorum.
Teşekkürler. Görünüyor herhalde, bir sorun yok.
Görüyoruz hocam.
Tamamdır. Ee, Bir de okay. Thank you for the kind introduction, and I would also like to thank for this invitation. It's great to be here. Today I will be talking about our recent chemiluminescent probes that we are using for different bio applications, mostly for in vivo imaging of different analytes. So in my lab at Koç University, we are developing small molecules. So I'm an organic chemist by training. So we are designing and synthesizing small molecule-based probes for different bio applications. So these are ranging from tumor-targeted phototherapy agents or sonodynamic therapy agents in which we are using ultrasound to trigger reactive oxygen species generation. And also some near IR emitting fluorophore again for bioimaging applications. And in some applications, we are combining these two approaches on the same molecule, so it can do both therapy and imaging at the same time, so-called theranostic probes. Today, I will be focusing on this part, basically, the chemiluminescent probes that we are developing for different imaging studies. If I need to give the simple definition of the chemiluminescence, so it can be simply defined as a reaction that emits light. So you are triggering a reaction, and as a result of this reaction, a light comes out. So the chemiluminescence imaging is quite promising and attractive compared to fluorescence imaging, because as you may see, there is no need for external light radiation. So what does it mean basically? So we can address the chronic problems of fluorescent probes, such as the limited penetration of the irradiation light, autofluorescence coming from the biomolecules. So these are quite problematic issues in the case of fluorescence imaging, which decreases the signal to background ratio. But in the case of chemiluminescence, so since there is no need for external light radiation, a reaction can take place in the cell, and as a result of this, a light is coming out, so there is no need for irradiation, so there is no autofluorescence coming from the biomolecules. We see a great example of chemiluminescence in nature, actually. So this is the firefly luminescence. What's happening in the case of firefly luminescence? So these insects contain firefly luciferin. This is the molecule. And in the presence of luciferase enzyme, a series of reactions taking place, and we are getting this excited state oxyfluorescin. And when this excited state oxyfluorescin comes back to the ground state, a light comes out. So this is a nice example from the nature. But of course, it cannot be applicable in human cells because we don't express luciferase enzyme. So the firefly bioluminescence is only applicable to in vitro assays. So why we are calling this bioluminescence? So it is basically under the title of chemiluminescence, but specifically we call it bioluminescence because it requires this enzyme to trigger the luminescence formation. So inspired by this phenomenon, basically, chemists years ago developed some chemiluminescent probes, so meaning that with a pure chemical reaction, light can be generated. So very first example is based on this luminol molecule. So when this luminol molecule is oxidized under alkaline condition with hydrogen peroxide, and again, as a catalytical amount of enzyme is needed in this case. So excited state species is generated, this aniline derivative, and then when it goes back to the ground state, a blue light is occurred. But there are many problems with this first generation luminol-based chemiluminescent probes, such as the first thing is that you need an oxidizing agent, such as hydrogen peroxide. It works very well under high pH conditions, so it requires alkaline condition, which is not fitting with the physiological pH, which is, as you know, 7.4. And the outcoming luminescence is in the blue region. So this is again a problem. And the luminol itself is having a poor water solubility. Again, it is not suitable for the bio applications. Then this 1,2-dioxetane-based chemiluminescent probes came out. So these are known as sharp dioxetanes. So Sharp is the guy who invented this structure. So what I mean by dioxetane is this four-membered ring here. Okay, so we have oxygen-oxygen bond. This four-membered ring is so-called dioxetane, and it is quite unstable. So in order to make it stable, a bulky, sterically hindered adamantyl group, which I'm highlighting here, is attached nearby in order to make it stable, at least to some extent. And again, on the left-hand side, we have this ring, benzene ring with hydroxyl group. So how it works, when this hydroxyl group, OH group, is masked, or you can call it caging unit or masking unit. So when I cage the phenolic hydroxyl group, the compound is stable and I don't see any luminescence. Okay, so there is no luminescence coming out. When I selectively remove this masking unit in tumor cells, for example, okay, with a tumor-associated input, such as enzyme, biotiols, small molecules. So when I remove this cage, chemi-excitation starts, and I generated this excited state benzoate. And then again, the same idea, when it goes back to the ground state, I see a luminescence. But again, there is a problem because the outcoming luminescence is having very low intensity in physiological conditions in aqueous solution at pH 7.4. Because the major problem here is that, so you need to generate this phenolate, okay? So after removal of the masking unit, caging unit, the resulting OH group should be deprotonated to generate phenolate. And the pKa of this hydroxyl group is higher than higher than 7.4, so the effective deprotonation is not taking place. So the logical idea here, which is done in 2017, after like maybe 30 years later, so if we can place, so this is the same structure of dioxetane, if we can introduce an electron withdrawing groups at the ortho position of this OH, we can decrease the pKa of the resulting phenol, and the phenolate concentration under physiological condition will be higher. So again, the same idea. I have the adamantyl, dioxetane here, masking unit, but with an addition of conjugated electron withdrawing group. So it can be acrylonitrile, methyl acrylate, acrylic acid, okay, something that can withdraw the electron density, and it should be conjugated to the ring. So what's happening? Again, it is stable initially. Remove the masking unit. You generate effective phenolate at pH 7.4. Chemi excitation starts. Dioxetane is broken. Adamantonon is released. So adamantyl group is departed. And then finally, I reach to this excited state species. Again, when it goes back to the ground state, I see luminescence. But this time it works perfectly in the aqueous solution at pH 7.4. So it is applicable in cell studies, applicable in vivo studies, in animal models. So by this simple modification at the ortho position of the phenol, the fate of the chemiluminescent or dioxetane probes has changed basically. And this, the 1,2-dioxetane structure that you are seeing here is quite modular. As I told you, it works at physiological condition, and also you can tune the luminescence wavelength. How? For example, in this pi conjugation here, if you extend this pi conjugation, basically you can shift the outcoming luminescence to the red region or even to the near IR region. Initially, with the single modification, it emits green light around 550 nanometer. But as I told you, if you extend the pi conjugation, you may have red emitting or even near IR emitting chemiluminescent probes. And also you can tune the luminescence kinetic by changing the stabilizer. So instead of adamantyl, if you use a smaller stabilizing unit, such as cyclobutyl, for example, the chemiluminescence kinetic rate will change. So I can show you a video here. Let me see if it works. Yeah. So in the case of adamantyl, as you can see, we have a long-lasting chemiluminescence. But when I have small unit, cyclobutyl unit, it is very intense, but the luminescence kinetic rate is very fast. So you can also, based on your end application, you can tune the luminescence kinetic, which is nice. Because sometimes you may need very fast reaction or very fast detection. So based on this new generation dioxetanes, we use it for many different applications. So in one of our earlier studies, by using 1,2-dioxetane probes, chemiluminescent probes, we targeted to detect carboxyesterase 2 enzyme in cancer cells. So what is the role of this carboxyesterase 2 in the cell? So it basically triggers the hydrolysis of ester, amide, carbamate, and thioester containing substrates. And what is more critical for us? We know that it has high expression in hepatocellular carcinoma. So you can treat it as a biomarker for hepatocellular carcinoma, such as, for example, liver tumors. Okay. And the problem with the carboxyesterase 2, so it has an isoform which is carboxyesterase 1. So it is very difficult to differentiate carboxyesterase 1 and carboxyesterase 2 with regular probes. Okay, so this is basically the challenging point here. So what we did, so we synthesized this dioxetane core. Again, we have the dioxetane here, adamantyl, ring, and this blue and red region is basically the masking unit, and we have the acrylonitrile as the electron withdrawing group. Okay, so in this form, there is no luminescence. It is in off state, nothing to see. Okay, and then when this amide bond on the masking unit, on the caging unit, is cleaved selectively by carboxyesterase 2, what we generate is basically aniline derivative, and then 1,6 elimination further triggers the release of the masking unit. And finally, we have the phenolate, as you can see here, and the chemi excitation starts after this removal of the caging unit, and you know what's happening after chemi excitation. We have the excited benzoate, and eventually we are seeing the luminescence. So this probe CS2 can only emit light in the presence of carboxyesterase 2, okay, because this amide bond is highly selective when we have aryl amides. These are highly selective for carboxyesterase 2 enzyme. So if you are interested in organic synthesis, I can briefly touch on that very briefly. So we are starting with this aldehyde, which is commercially available. And then we initially form the acetal, and then TBS protection of the hydroxyl group, followed by the phosphonate ester synthesis. And then we attach LDA, in the presence of LDA, we attach two adamantonone. So we have the adamantyl group here, and then regular deprotection of the TBS to go back to phenol. Monoiodination with an iodosuccinamide gives orthoiodinated product, and Heck coupling after this step is basically generating this enol ether. So why I'm saying this enol ether? So because here this is the enol ether structure, and we don't have the dioxetane yet. On the first row, you see the synthesis of the masking unit, and then we took this enol ether, which I showed the synthesis in the previous slide, combined with the masking unit. So I attach my masking unit here, and the final step is the critical step. It is basically the formation of dioxetane. So how we form this dioxetane ring? We take the enol ether. Okay, so this is the precursor of the dioxetane. We add methylene blue. Methylene blue is a very well-known photosensitizer, and when we shine light, it generates singlet oxygen. So this in situ generated singlet oxygen reacts through cycloaddition reaction to this enol ether, and we form the dioxetane, which is basically the final product. So in the last step, basically, we are generating in situ in the flask, generating singlet oxygen, which is reacting with the enol ether structure here to form the dioxetane. So with the probe in hand, of course, we first check its response to carboxyesterase family. What I mean by the family, we use 1 and 2, carboxyesterase 1 and carboxyesterase 2 in solution. Okay, so these are in solution experiments in aqueous solution. So as you can see, when we treat the probe with carboxyesterase 2 blue curve, time-dependent luminescence turn-on response. So we have a huge increase in the luminescence intensity. A plateau is reached, and then gradually it decreases over the time. So in approximately in 30 minutes, we reach the highest intensity. And critically and importantly, when we use carboxyesterase 1, which is very difficult to differentiate from carboxyesterase 2, green curve, it's not even seen here because there is no luminescence. So the luminescence cannot be activated in the presence of other isoforms. And without enzyme, the probe itself is also stable throughout the time if you follow the red curves. And also, when we check the outcoming luminescence wavelengths, as I mentioned, since we are using the green emitting one, it is around 550 nanometer, and we were able to see more than 200-fold turn-on response in the presence of carboxyesterase 2. And then we check the enzyme concentration dependence. Of course, as the enzyme concentration increase, we see more signal. And when we treat with several inhibitors of the enzyme like LPA, BMPP, so these are different inhibitors for the carboxyesterase family, we see a decrease, suggesting that the outcoming signal is originated due to the carboxyesterase 2. And then when we check the luminescence in cell studies, so we took 3 different cancer cells. These first 2 are pancreatic cancer cells, and HEP2 is the liver hepatocellular carcinoma. And we first checked the protein level of carboxyesterase 2. As you can see in the Western blotting, we have huge expression in the case of liver cancer, and this is also reflected in the chemiluminescence imaging. We see the high signal in the liver cancer. And again, in the cell studies, we have the high signal with HEP2. When we treated with the inhibitor, we lost the signal as expected. So this is one of the first examples in which we use the 1,2-dioxetanes as the chemiluminescent probe for cancer cell detection. And then we thought that, okay, so we dive into the more details with the esterase family, and we try to image this time butyrylcholinesterase activity in cancer cells. So this is another esterase enzyme. This time it catalyzes the hydrolysis of ester molecules such as acetylcholine. So butyrylcholinesterase is quite important. It has like unique roles in physiological processes, but when there is a change in its level overexpression or low expression, it may be associated with the pathogenic states such as neurodegenerative disease, depression, liver damage, which is also correlated with the insulin resistance. And also it affects the fat metabolism, cardiovascular disorders, and importantly also we may see it as a biomarker in tumor formation or in cancer cells. So it is very critical to image selectively butyrylcholinesterase activity in real time in cells or in vivo. So this time what we did, again, we have the same core structure, electron withdrawing group, dioxetane, adamantium as the stabilizer, and on the ring, of course, we need to design and change the masking unit according to butyrylcholinesterase activity. So it is esterase, it cleaves the ester unit, but the R unit of the ester is quite important in order to achieve selectivity. And we found that when we have this cyclopropyl group, it's also reported in some other literature examples. So when there is this small cyclopropyl unit, this ester unit can be specifically cleaved by butyrylcholinesterase, but it cannot be cleaved by the other competing enzyme, which is acetylcholinesterase. So here the challenge is again to selectively reach butyrylcholinesterase imaging, but we shouldn't have any response from the acetylcholinesterase because acetylcholinesterase has some other roles, so we need to differentiate them. The conventional method that detects the activity of butyrylcholinesterase is known as Elman method, and unfortunately, Elman method cannot differentiate between butyrylcholinesterase and acetylcholinesterase activity. So that was our aim. So we wanted to generate highly selective chemiluminescent probe for butyrylcholinesterase. Again, when butyrylcholinesterase cleaves this blue masking unit, we will have the phenolate, chemi-excitation starts, and we see green luminescence. When we check the synthesis, so I have already shown you the synthesis of this core structure. Through esterification reaction, we attach our masking unit, and then in situ generation with the methylene blue, oxygen, and light, we generate signet oxygen, we form the dioxetane. Again, in solution, let's see what we obtained. Time-dependent luminescence detection. As the enzyme concentration increases, we are seeing higher signal, and almost in like 10 minutes, we reach to the maximum point, and it decreases gradually as the time passes, and after like in 2 hours, almost quenched. And when we add tacrin, which is the selective inhibitor of the butyrylcholinesterase, we lost the luminescence. This is a highly critical data because here we check the selectivity, and as I mentioned, there are two main competitors, acetylcholinesterase and carboxylesterase 2. Remember, again, this is esterase, but we only see huge luminescence intensity increase in the case of butyrylcholinesterase, and negligible signal came out in the case of other esterase enzyme. And in cell, what did it do? So we took neuroblastoma cell and liver cancer cell, which overexpress butyrylcholinesterase, and normal cell line HEC293T. And as you can see in green and red bars, we have higher luminescence intensity increase in the cancer cells, but lower in the normal cell line, so it can differentiate between normal and tumor cells. And again, when we treat the cells with tacrin, the luminescence intensity decreases. And here we also wanted to check in vivo tumor imaging. So we generated a tumor on the left flank of the mice. Here you can see the tumor formation with neuroblastoma cell line. And then when we injected our probe, here you can see highly selective imaging coming at the tumor site with a very high signal to noise ratio. So there's no background basically. because it can be only activated in the tumor region. And again, as the time passes, we see increase in the luminescence and then gradually decrease. Okay, so after dealing with this esterase enzyme, so we thought that now maybe it's time to play with the core structure of the dioxetane. So we thought that, okay, chemiluminescence imaging is working very well, so we can design the cage units, we can target different enzymes, but how about can we control the subcellular localization of the dioxetane? So when it enters the cell, can I control where it will localize? So organelle targeting is quite important, as you may know. So because if you have like specific probe that can localize to a specific organelle, you can precisely control the trafficking, signaling pathways, and organelle related processes. So it gives you huge info, and this is quite important to understand many different things. Then we thought that with simple modification on the core structure, can we design the first example of organelle targeting chemiluminescent probe, and we targeted mitochondria initially. So in order to target mitochondria, the common approach, the most popular approach, is to have cationic units, because we know that the mitochondria membrane potential, so that there's transmembrane on the mitochondria, and the membrane potential is negative, and it is even more negative in tumor cells. So somehow, if you can design cationic dioxetane, it will preferentially located on the mitochondria. So in this direction, again, we took our dioxetane, adamantyl here, benzene ring. So we have electron withdrawing group. But on top of electron withdrawing group, we further modified it, and we attach this alkyl unit with a triphenylphosphonium group at the very end of the molecule. So triphenylphosphonium, so the phosphorus is positively charged, so it makes the probe cationic, and it will allow us to target mitochondria. Triphenylphosphonium, by the way, is a very well-known mitochondria targeting unit. It's been used over the years for targeting mitochondria with fluorescent probes. But this is the first time that we implement this approach to the chemiluminescent probe. And then what is the targeted enzyme? It is basically leucine aminopeptidase, LAP in short. So LAP is a quite important biomarker. It is overexpressed in several cancer cell lines, such as lung, liver, colon. So it is quite important as a biomarker. And what is the job of leucine aminopeptidase? It hydrolyzes the N-terminal of leucine from peptides or proteins. So as you may guess, the strategy should be, so we have this leucine amino acid attached to the self-immolative linker through amide bond. Okay, so LAP enzyme will recognize leucine amino acid, so it will selectively cleave this peptide bond, amide bond, in other words. Again, we will have free amine, 1,6 elimination, so it will be triggered by itself once you generated the amine. Electron movement, 1,6 elimination, will further release this linker, and at the end of the day, we have the desired phenolate at pH 7.4. So upon formation, this phenolate, again, chemi-excitation starts. We have the benzoate excited state, and then luminescence coming out. Okay, so the chemi-excitation part is the same, as you can see, but this time we will be localizing to mitochondria, and we will image mitochondrial LAP enzyme activity. And since this study, the relation of LAP enzyme and the mitochondria was not fully understood, so it basically also helped to understand in tumor cells we have high LAP activity in mitochondria. If we check the synthesis, so basically again with this core structure, we first got the NHS ester, and then amide coupling reaction will give us the triphenylphosphone unit at this end, and then FMAC protected leucine conjugated to the ring, so masking unit is here, Classical deprotection of the FMAC unit with piperidine in DMF gives the leucine itself. And then in situ generation of signet oxygen. Signet oxygen is added to this enol ether, and we have the dioxetane. So this is MCl is the final probe. Then we took MCl and tried to see the leucine aminopeptidase triggered luminescence intensity increase in different cancer cell lines. HCT116 is basically a colon cancer, HG2 you know liver cancer, A549 is the lung cancer. So in all of them, as the time passes, we see a huge increases and then gradual decreases as expected. And in all these three different cancer cell lines, we were able to see the leucine aminopeptidase activity. But the highest intensity was observed in liver cancer, and which is, as you can see here, the total luminescence is high in the HG2. When we use fibroblast normal, non-cancerous, normal fibroblast cancer cell line, no luminescence was observed. So this is critical in order to differentiate between tumor cells and normal cells. And when we treat HG2 cells with bestatine, again, the non-inhibitor of the LAP enzyme, a dramatic decrease in the luminescence was observed. And as I told you, we want to control the subcellular localization of our probe. So the co-localization studies were needed. So here you can see the green signal coming from our chemiluminescent probe from MCL, and red coming from the mitotracker red. So this is commercially available known mitotracker dye. So if we need to see a nice overlap between them, and as you can see in this zoomed in version, the green and the red emissions, the signals are perfectly overlap, suggesting that the high mitochondria targeting ability. And also, it worked very well in the in vivo. So as you can see here, we have this two tumors at the left and right side of the mice, and when we injected MCL, our probe, we see a high intensity in the tumor region. And when there is no tumor, so these mice are having no tumor, so these are healthy mice. And when we injected the probe, no luminescence came out, which is critical for us. And then by using this mitochondria targeted scatterland, the core structure, we wanted to catch hydrogen sulfide activity in different biosamples. So in order to do so, this time we simply changed the caging unit. So we have this dinitrobenzene as the masking unit, okay? And the core structure is same. We have triphenylphosphonium as the mitochondria targeting unit, dioxetane here. In the presence of hydrogen sulfide, the SNR reaction is taking place and chemi-excitation starts. We have the luminescence. Hydrogen sulfide, as you may know, again, it has critical role in physiological conditions, but abnormal levels are quite critical for different disease, such as, again, cardiovascular, diabetes, neurodegenerative disease, and cancer. So in colon cancer and neuroblastoma cancer cell lines, we see time-dependent luminescence turn on, which is nice. Here you can see the mitochondrial localization. Again, green is coming from our probe, red from the commercial tracker. We have high Pearson correlation coefficient, suggesting a nice overlap. And when we add zinc chloride, which is basically the quencher of hydrogen sulfide, we see a dramatic decrease in the luminescence intensity, which is again nice to show that hydrogen sulfide is doing the job. So here we wanted to apply another strategy and another application. And for the first time, we detected hydrogen sulfide in human serums. So from the Koç Üniversitesi Hospital, we collected 10 different healthy individual serums, 6 female, 4 male, aged between 22 and 40. And as you can see here, as the serum ratio increases, we see a huge intensity increase in the luminescence. And again, treating it with the zinc chloride quencher, it decreases the luminescence. And also when we add sodium hydrogen sulfide to the serum, so when we boost the hydrogen sulfide level in the serum, we were able to see again further increase depending on the concentration of the added sodium hydrogen sulfide. So it means that in the serum, our probe, in the real human serum, the probe can detect the changing concentration of the hydrogen sulfide, which is quite important. And here you can see the 10 different human individual serums. And since these guys are healthy, so we have more or less the same level of hydrogen sulfide. And again, treating it with the quencher, we see a decrease in the luminescence. So this is quite important because it is showing that the chemiluminescent probes can also work in the human serum. And the next application would be detecting different biomarkers in human serum for the diagnostic purposes. So these were just like proof of principle study. We worked on the healthy individuals, but it can be done in the disease state. So you can collect serum samples from the patients, and you may want to detect the activity of different biomarkers. And in the last part, I want to show you another application, different application with the chemiluminescent probes, in which we developed chemiluminescent nanoparticle this time, which can emit light for a very long time upon a short irradiation. So this phenomenon is known as afterglow luminescence. So afterglow luminescence, once more, basically means you shine light, you excite your material, turn off the light, and monitor how long it can emit light when the excitation source is turned off. Okay, and this afterglow luminescence, if it is long enough, especially, can be used in the long-term cell tracker applications. And why these long-term cell trackers are quite important? Because it can give you this opportunity so you can monitor cell proliferation and migration, cell-cell interaction, even also metastasis, immune response, and more importantly, you can run image-guided surgery applications because if you have a very long emitting material, so it can allow you image-guided surgery. So how we designed our afterglow chemiluminescent nanoparticle, what are the components of this nanoparticle? Let me first introduce them. So we have this methylene blue derivative with a huge hydrofluoro tail, which makes it hydrophobic. So we have hydrophobic version of methylene blue as a photosensitizer. Okay, so this is as a photosensitizer. And also we have this enol ether. So please remember from the synthesis. So when you have this enol ether, you don't have yet the dioxetane. So this is dioxetane precursor. In order to be dioxetane, it needs to react with signet oxygen. Okay, so it's not dioxetane yet. We took these two hydrophobic molecules and mix it with amphiphilic polymer, DSPE PEG, containing cell-penetrating peptide, TAT, at one end. Okay, so this is amphiphilic polymer with cell-penetrating peptide. So when we mix these three, a nanoprecipitation takes place, and basically we are forming a micellar structure. So this amphiphilic polymer covers these two hydrophobic unit inside, okay, and the hydrophilic part of the polymer stays outside, interacts with the water, okay. So we form a micellar structure. Our two hydrophobic units are inside the polymer. Then we shine 660 nanometer laser. Why 660 nanometer laser? Because methylene blue absorbs at that range. So we basically excite our methylene blue. Methylene blue is a photosensitizer. Under light radiation, it will generate signet oxygen, and this signet oxygen will react with the other component, which is the enol ether. So in situ, we will generate this dioxetane. And since this nanotorch, we call it nanotorch, this micelle nanotorch, is in pH 7.4, immediately this hydrogen hydroxyl group will be deprotonated. So we will generate phenolate and chemi excitation will start. Okay, so this is the idea. So we will just shine 660 nanometer laser. I will turn off the laser, and then I will monitor the afterglow luminescence coming from my chemiluminescent dioxazine. Here you can see the TM images of the nanotorch, this my cell, with a size of around 190 nanometer. And then when we have the laser, near IR laser light, and our nanotorch, we only see the outcoming luminescence. So these are taken by the IVIS imaging system. And as the concentration of the nanotorch increases, we see more luminescence upon laser irradiation as expected. So this is basically the most critical part to show how long the afterglow luminescence is taking place. So we took our nanotorch, we irradiated for 5 minutes with 660 nanometer laser. We turn off the light and then monitor the outcoming afterglow luminescence. In aqueous solution, as you can see, so this is where we turn off the laser, and you see the afterglow luminescence lasting for 10 days. So after 5 minutes radiation, we have a material that can emit light for 10 days in solution, which is a very long, huge number in the field of afterglow luminescence. So this is, of course, in solution. And what is more, you can recharge this afterglow luminescence as it goes down. You can shine the light laser. Again, you can recharge it. Okay. So, of course, we are using more dioxazine ratio compared to methylene blue in order to recharge it. And the outcoming luminescence is around 570 nanometer. What can be done with this very, very long afterglow luminescent material? So basically, we design a cell-based micro-robot. So what does it mean? We took microphage cell, incubated our nanotorch, my cell, and in addition to that, we also incubated the cells with iron oxide nanoparticles. Okay, so iron oxide nanoparticles are needed because we want to control the migration of the cell from outside with external magnetic field. Okay, so that's why we call it basically cell-based microrobot. Again, in the cell, we shine the light for 5 minutes, turn off the light. It emits for more than 2 days in cell medium. Under confocal microscopy, you can see the luminescence, and also under confocal microscopy, you can see these small iron oxide nanoparticles. The nanotorch itself is not toxic to the cell, which is nice because I want to do just imaging, so it shouldn't be toxic. And then what we do, so we took this cell-based microrobot and applied external magnetic field. So in the influence of magnetic field, cells are migrating, and thanks to this very long afterglow, we were able to image this migration. So in this video, you can see we are applying the external magnetic field. These small cells are emitting afterglow luminescence, and because it is lasting forever, we were able to see the migration. So can it be also done, or is it gonna work in vivo? So this what I would like to show now. But before diving into this application, I would like to show you the advantages of afterglow luminescence, or in general chemiluminescence versus fluorescence. So as you can see in the chemiluminescence mode, we don't see any background, but when we do the same imaging with same material via fluorescence mode under IVIS imaging system, we see a huge background signal. So this is a critical figure that is basically showing the advantages of the afterglow luminescence. Again, in vivo, it is rechargeable, and how long it lasts in vivo, in animal. So of course, the degradation is slightly quick in the case of in vivo, but still it can emit for 1 day in animal model. So again, which is quite useful and advantages. And finally, as a proof of principle to show this, what we did, so we took our cell-based microrobot containing nanotorch, iron oxide nanoparticle, macrophage cells. We injected them here. So of course, they start to emit. And then we place the magnet to this green highlighted region. So the cells are migrating towards the magnet. And again, thanks to long afterglow, we were able to image this migration. And then when we remove the magnet to the left side, to this red highlighted circle, cells are moving there as the time passes, and the material is still luminescent. So that's why we were able to image them. So as you can see, with external manipulation, we can migrate the cells, and this migration can be imaged thanks to afterglow, long afterglow luminescence. With that, I would like to conclude my talk by thanking my research group, my students basically, who did all of these studies. So without their effort, none of them can be done. And I have like many great collaborators. So I just listed who contributed the studies that I mentioned today from China, University of Toronto, and from Koç University Hospital. And of course, I would like to thank the funding sources and all of you for being here. I will be happy to answer any questions that you may have. Thank you.
Thank you, hocam. Now we have a Q&A session. You can directly speak up or you can use chat box.
I have one question, actually several questions.
Sure, hocam.
It was a very nice presentation, thank you. I just wonder, I'm not in this area, but it was very interesting, and I wonder if you have used your nanoparticles for other applications or your molecules for other applications rather than detection and diagnosis.
Thank you,
This is
hocam.
one question. The second one, you propose that the irradiation, the conventional chemiluminescence samples are irradiated, and you showed it as a disadvantage. But at the end, you synthesized chemiluminescence nanoparticles upon with irradiation.
Which requires irradiation.
Yes. Why did you choose to synthesize that if this is a disadvantage?
Yes,
And
thank
one
you.
more, you also used to make this molecule nanoparticle fluorinated structures, which is very dangerous even for energy applications. And you are using it for diagnosis. And I don't know, are you injecting or what kind of application you make it? You directly send it to the body, in this case for the mouse, right? If you use it for human beings, it's accepted as a very toxic chemicals, even for battery applications. I know it is hydrophobic. It's easier to make it to synthesize these structures. Yes. Thank you.
Thank you, hocam. Like all of them are great questions. So let me start from the first part. Currently, we are also trying to implement therapeutic action to this nanotorch. So if you can add a drug, and for example, you can take your micelle again with the magnetic field to the tumor region, so you can both image and induce therapy at the same time. So basically, we are now including a drug in the micelle in order to also have therapeutic action on the tumor site. So this is one example, for instance, so what we are working on. So regarding your second question, you are right. So I first mentioned like I don't need excitation, but then I use excitation. But in the case of this nanotorch for afterglow luminescence, there is one advantage which basically removes all disadvantage of the external light radiation. We can take our nanoparticle, excite them outside the cell or outside the animal, and then inject them to the animal. So basically, we charge them outside the cell or outside the animal and then inject them. This long afterglow luminescence gives us this opportunity. Since they emit like for one day, I can excite them outside, I can charge them, initiate the luminescence, and then I give to the animal. So that's why I basically bypass all drawbacks of the excitation light usage. So that's why we are using light basically. Otherwise, you are right. So if I again use light on the animal, that would be disadvantages. And when it comes to your third question, If
Fluorinated
I
structure.
remember, if I remember.
Fluorinated, fluorinated structure.
Fluorinated, okay. The fluorinated one, yeah, so you are right. But as you know, these are all depending on the dosage, right? So we are using very small amount of fluorinated methylene blue, and we also separately check whether it is toxic to the cell without before using it. So in the concentration range that we are using, they are not toxic. But you are right, of course, if I need to increase the concentration, it might be toxic. So we are taking care of this safe concentration range while using any kind of photosensitizer, basically.
Yeah, it is to make the structure is hydrophobic.
Yes, yes, yeah.
But there are other options.
Yeah.
To
So
make
we can use other hydrophobic photosensitizers, as I told you in the last part. So for example, we have iridium cyanine, and also for that one, if I use high concentration, iridium would be toxic as well.
Yes.
So these are all related with the concentration, and of course we shouldn't decrease the ROS generation. High signet oxygen generating photosensitizers are needed. But of course, more safer versions might be chosen.
I may ask one more question.
Yeah, sure.
The last molecule was long-lasting one, if I'm not mistaken.
Yeah, slightly longer.
Can we use it to determine some sort of materials lifetime test to follow to detect the lifetime of the materials?
Yeah, you can do it. So you can basically tune the polymer that you cover them for different applications. So at this moment, I don't know how can I do this, but it is very modular. So you may apply it for different applications. So it is quite possible.
Okay, thank you.
Thank you.
Safacan hocam, sorry. Related to Tülay Hoca's question about the toxicity, what is the highest toxicity that you checked the cell death?
I need to double check it, of course, but for sure it is in micromolar concentration. So I would say maybe like 50 micromolar or so. So it's around
But
those
is it
range.
too much? I mean, do you think that is it too much, 50 micromolar?
Because we are using in the range of 5 to 10 micromolars. So it is like for us too much, but maybe in general not. But since I'm using very small amount of photosensitizer, so let's say I'm using 5 micromolar, but I'm checking like 50, 100 micromolar.
Okay.
To
You use it in 5 to 10
Yeah,
micromolar range, but the highest one is
maximum
the
10.
50, 100 micromolar.
Yeah.
Okay. Can I ask second question?
Yeah, sure.
I don't know, maybe I missed. I saw you target the mitochondria and you compare it with the mitotracker.
Yes.
Have you ever tried to target the lysosome in a similar way?
Yes, we did. I just submitted the manuscript.
Okay. Okay.
So it also works.
Is it similar or?
Yeah. So the nice thing with this core structure by chemical modification, and
Okay.
since I'm organic chemist, so we can synthesize like many different modified versions. So rather than triphenylphosphone unit, which is cationic, I can simply change it to morpholine, for example. So morpholine is gonna take us to the lysosome. And also I can do it ER, Golgi, so whatever you like, I can synthesize actually. So it is very modular and chemistry allows it.
So your chemical structures, bind to the lysosome surface, right? Is it a covalent binding?
So it is kind of interaction, electrostatic interaction. Yeah, because you know that the acidic we are taking use of the acidic environment of the lysosome.
Yes,
So
this
I'm
is
adding,
why I ask it.
I'm adding basic unit to my core structure, and I'm making use of the acidic environment of the lysosome. So it is like different charges again attracting by themselves. So it's electrostatic basically.
Is it
Not
possible?
covalent.
Is it possible to conjugate any drug or any small molecule to your system. That, I mean, after targeting this drug, able to enter the inside of the lysosome?
Yes, you can do that.
Okay.
So we are not specifically we are doing for the lysosome, but we have dioxetanes also carrying drug for different applications. But as you mentioned, you can also implement organelle targeting drug on the dioxetanes. It can be done.
I'm specifically asked to lysosome because it's hard to target and it's hard to actually carry the drug inside of the lysosome because of the acidic environment.
Yeah. You are right. So beside this chemiluminescent probes, we have some lysosome targeting photosensitizers as well.
Okay.
So because like half of the group is also working on the photodynamic therapy. So we have photosensitizers, which is activity based again. So for example, it is getting activated with lysosomal enzyme, plus it is targeted to lysosome.
Ah, great. Okay.
Yeah, all of those are possible.
Okay. Okay, thank you very much. And
Yeah, thank you.
it was a very nice presentation, by the way. Thank you.
Thank you. Yeah, thanks for coming.
Any question? Safacan Hocam çok teşekkür ederiz. Çok güzel bir webinar oldu.
Sağ olun, ben de teşekkür ederim.
Vaktiniz için çok teşekkür ederiz. Değerli katılımcılar, size de katılımınız için çok teşekkür ederiz. Yeni bir webinarda görüşünceye dek hoşça kalın demek istiyorum. Tekrar teşekkürler.
Nokta Hanım.
Buyurun hocam.
Hocam, Safacan Hocam, sizden bir miktar örnek alıp denemek mümkün mü burada? Ben bir şey de denemek istiyorum.
Tabii hocam, şey yapalım, mailleşelim, konuşalım.
Tamam.
Olur tabii ki.
Çok teşekkür ederim. Çok farklı bir uygulama.
Sağ olun,
deneyeceğim.
memnun oluruz.
Oldu. Görüşmek üzere.
Tamam,
Teşekkürler
görüşürüz.
hocam. Hoşça kalın.
İyi günler, kolay gelsin.
Evet,
Sağ olun.
teşekkürler.